and welcome Mindsets to this session of Learn Extra Live, grade 12. Hope you guys are ready. You've got your pens, pads out, and you're going to make some notes. I'm Ty, and I've got Aslam here who's going to be taking us through today's session. Aslam, what are we doing today?
We are tackling paper one because the learners are writing paper one on Friday. That's the whole of the exams, insignificant subjects are over. The big one starts on Friday. The whole year it's got nothing to do with anything else. Life science is the thing. We're doing genetics and other stuff from paper one.
All right. Okay, so while Aslam makes his way to the board, Mindsetters, you know the drill. You need to get onto the Facebook page, www.facebook.com/learnextra. Chat to me, let me know what you guys are thinking. If you're lost in it, if you need help. I know I better see this page exploding because you guys are writing very soon, so make sure you get chatting to me. Whatever question you have regarding this paper, make sure you post it up and in detail so I can be specific. But on that note, this is where I hand over to Aslam. Aslam, take it away.
Thank you, Ty. Welcome, Mindsetters. I'm hoping you focusing on life science now. The last couple of weeks, we noticed you were quiet, obviously, because you were learning for other subjects. Now we're moving towards that stretch where we're preparing for life science. And remember, guys, if you prepare properly, you're writing a preparatory exams in Gauteng. We're talking about Gauteng writing on a Friday and next week, Tuesday. Other provinces may be writing at other times. But remember, the preparatory exams, guys, to prepare. If you prepare well now, your preparation for end of the year becomes much easier. It just becomes revision again. So please be with us, be on the page, give your questions. We would stop at any time to take some of your questions which you feel are very important. So just keep those questions rolling. Just to go back, we're talking about paper one, and the section that we haven't covered well is the section on genetics, and this also from your request, genetics and biotechnology, and genetical modifications, etc., etc., etc. Just to, as our plan has been in the last couple of weeks, we start off by talking about what is the section all about. Obviously, genetics. First, we talk about concepts in inheritance, and from there, we talk about Gregor Mendel, the guy that has given us so much insight into the section, and we look at his laws of dominance and law of segregation. We also want to know all the terms. And guys, in this section, you've got to know the terminology. If you know the terminology, the work becomes easier. If you do not understand terminology, for example, if the question says a homozygous white bull was mated with a heterozygous black cow, and you don't know what homozygous and heterozygous means, that means you cannot proceed with that problem. You're stuck at the red robot, and you can't proceed there. So you need to know your terminology to negotiate the problems that you will encounter. Also, we looked at mutations here as well. And guys, in this section, the mutation goes back also to what we've done in protein synthesis and the rest of it. So there's a link between the mutations that we spoke about earlier. Then we go to the core of the section, and that is the problems, genetic problems. And here, fortunately for you, for this year, and next year, grade 12s, those that are writing version 1, remember, not those that are writing version 2. Those that are writing version 1 life science. That means full-time candidates that are writing for the first time life science. You only have to know about monohybrid crosses. That means you look at one characteristic at a time. But let's look at what we have to look at. Complete dominance, incomplete dominance, codominance, inheritance of sex, inheritance of hemophilia and color blindness as sex-linked characteristics. And we look at other characteristics as well. Then we want to know whether you can interpret a pedigree diagram. Lastly, we look at genetic engineering, ethics, and legislation. That forms the last part of the section. Here we look at how we use genetics in agriculture, how we use genetics in medicine, how we use genetics in genetic counseling. That is, if you want to get married and you have a genetic disorder, you want to know from a counselor what are the chances of getting a baby that has this problem that you have, etc. You get all of that. Also knowing selective breeding, and remember the section on selective breeding is linked here and in evolution later in paper 1. And lastly, you look at cloning. And guys, cloning you've already covered in grade 10 when you did mitosis, and we've covered it under meiosis as well, and under DNA we've looked at cloning, what it means to make a copy of an organism, an exact copy of an organism. That's what we mean when we talk about cloning. So that, guys, in a nutshell, is what we need to do. Now, let's not linger on this. You have it there. It's on the page. You can look at it. And also you have what we call Circular E16 of 2010, which has the exam guideline. So please consult that to tick as you go along what you have done. Now, guys, we move straight into the question time. The first set of questions that I have selected today are to revitalize, or to recap, rather, not revitalize, recap the terminology that is essential for this section. First one says, and look at the instructions, it says provide the correct biological terms for the following. Now, if you look at your paper, this is normally question 1.2 in any life science paper. Question 1.1 would be the multiple choice questions. Question 1.2 would be this correct terminology. Question 1.3 would be either matching or multiple matching, and so on the questions go. Here they're saying you must give the biological terms. Now, first of all, I didn't put all the instructions there. In your exam booklet, you're going to write down the numbers here, in this case 1.1.1 through to whatever number we have here, and next to each you will write the correct terminology or the term that fits the statement. You do not rewrite all of this. Smallest unit of heredity in a living organism found on the DNA, and that would be the gene. 1.1.2, genes controlling the same characteristic located at a specific position on a specific chromosome, that would be alleles. This now is, remember, we get characteristics we get from our parents, one from the mother, one from the father. So, for example, eye color, we're getting one gene from our father and one gene from the mother. So these two genes, they are controlling the same characteristics, eye color, and they would be found on this relatively same position on homologous chromosomes. I'm not going to go into details of homologous chromosomes. We've gone into that in meiosis. So they are called alleles. So you talk of a gene and its allele. One may be coding for blue eyes, the other one may be coding for brown eyes. Depending which one is dominant, that's the color of eyes you would have. Sometimes both may be coding for the same characteristic or the same color in this case. Let's look at further and we'll get some of those definitions. Gene that masks or covers the expression of another gene, that is the dominant one. So linked to Mendel's law of dominance. And I prefer to call this the cat and the mouse. When the cat is away, the mice will play. What does this mean? That the cat in this case is the dominant gene. And as long as the dominant gene is there, the weaker gene would not show itself. It will be there, but it would not show itself. A perfect example is eye color. Brown, if you have a gene for brown eyes and you have a gene for blue eyes. Brown is the dominant, so brown's the cat. And blue is the recessive, so that's the mouse. So as long as you got the brown gene, your eyes would be brown and not blue. Only when that brown gene is absent, then the blue would show up. So when the cat is away, the mice will play, right? Gene of which expression is masked or covered, that's obviously the reverse of this one, and that would be the recessive. A genetic cross involving only one characteristic, and in your case, that's easy, mono. Mono meaning one, hybrid cross. Monohybrid cross, the genetic makeup of an organism. That means, is this organism heterozygous for a characteristic or homozygous for this characteristic? Whether it's eye color, whether it's skin color, size of nose, size of ear, size of bum, whatever it is, but we are talking about the genetic makeup. That would be the genotype. Genotype. So genotype can either be heterozygous or homozygous. Physical expression of a characteristic, this would be for physical, for phenotype. phenotype, that means what color are your eyes. When I look at your eyes, are they blue, brown, green, yellow, orange? Today we don't know because you're wearing those different contact lenses and your eye color changes every day. You've got a color for every day. And there we go, 1.1.8. Condition when a gene and its allele are identical, as I alluded to just now. When the gene and its allele are identical, that means they're both coding for the same color in that characteristic or the same feature in that particular characteristic. Then we say they are homozygous. We can also say that the organism is true breeding for that particular characteristic. The organism is true breeding, as opposed to homozygous there. Condition where a gene and its allele are different, we use homozygous there, so that will become heterozygous, different. Or we can say hybrid, okay? When both alleles of a gene are equally dominant. Example, blood type. When they are equally dominant, none of the two are stronger. They are two different genes, but none of the two are stronger than the other one. When they equally, that means when they show up, like for example, you take white cows and you mate them with black bulls and you end up with a black and white cow. That means black is there and white is there. You don't get a new color, you're getting a color which shows both features. That will be they are equally co-dominant. A phenotype characteristic that is controlled by more than one gene all working together, that would be multiple alleles. Sex-linked genetic disorder characterized by blood that does not clot well. So the clue here, first of all, it's a mutation. Secondly, they tell you that the mutation doesn't allow blood to clot well, and this is hemophilia. A condition where the blood doesn't clot, and because the blood doesn't clot, even the smallest of cut can lead to death if not controlled properly. So that, guys, is our section on section A, and it helps you just to get those juices flowing so that you know the terminology. Now we're going into the problems. In mice, brown color is dominant. They're telling you it's dominant, and they're showing you that they're using a capital letter B for that. In genetics, we use a capital letter to indicate dominance, and we use a small, lowercase letter to indicate recessiveness. Show a crossing between a heterozygous parent with a brown coat color and one with a gray coat color up to the F1 generation. Also give the phenotypes of the F1 generation. Now guys, you notice I've underlined keywords here. One is, what is the dominant one? What is the recessive one? I didn't do that, sorry about that. And so on, and we go along that way. Now, you must know this terminology before you actually answer. So we're looking at brown, which is dominant, and gray, which is recessive. They want to know a heterozygous parent with brown. By the way, a heterozygous parent, according to this definition that brown is dominant, will always be brown. You cannot get a heterozygous gray, because when the cat is around, the mice can't play. Remember that rule. Now watch how we set this problem out. This is very key because you're getting marks for everything you're saying there. So we got a brown and we got a gray parent. Notice that's the phenotype. So we're giving the actual feature, brown and gray. The genotype, the brown one we said was heterozygous. So we're going to say B and small b. And the gray has to be two small b's, because a recessive characteristic can only show itself in the homozygous connection, in the condition, sorry. Because if it is heterozygous, that means the cat is there. And if the cat is there, the mice can't play, then we get back to this. This one here could have been either BB capitals or B small b. But they told you that it is heterozygous. So we can only use the heterozygous condition there. We go down with that. This meiosis, we write meiosis there. And so the gametes then separate. We have B separated by another one there, B, and those two separated there. Notice there's a space between them, or a comma, indicating that the gametes have separated. The gametes have separated because meiosis has taken place. Notice here on top we have P1. I want to see how far I can go with that. Okay. Now what we do, this is one parent. That's the other parent. So what we can do when fertilization takes place, we can either do that to combine with that one there, we get B and small b, and we can do that and that, we get the same thing there. Then we do these two, small b, and that one, small b, small b. You notice the diagram is a bit cluttered. So we'd rather use a Punnett square where we keep whatever is on the left-hand side, and whatever is on the right on top in this case. The idea is to separate them. That one and that one gives you capital B, small b. That one and that one gives you capital B, small b. These ones will give you small b, small b, small b, small b. So that is our answer for this. We'll take a little more discussion on this just after the break.
Yes, on that note, wow, that's a lot of information. For Mindsetters, make sure you keep chatting to me on the page. I'm loving it, and literally my computer is about to melt because you guys keep on posting, which is awesome. So make sure you guys keep posting. But on that note, we're going to see you after this break. And welcome back, Mindsetters. Hope you had a nice little break. Now you're back, you're ready, you got your pens, pads out, and you're going to make some notes. And by the way, Aslam, actually, I had a question coming from Spamandla. He wanted to find out, can you please explain what is meant by polygenic inheritance?
Spamandla, polygenic inheritance. Let's just do that very quickly. Poly, as you can see here, poly means many. Okay? So polygenic, many genes controlling. So first of all, there are more than 2 genes controlling the characteristic. That's one. More importantly, in polygenic inheritance is that the result of this, because of the variety of genes that control it. For example, if you take skin color ranges from dark to light. We don't have a specific either. You have, as we know, so stereotyping, saying you're either black or white or whatever. We don't have that. We have ranges in between black and white. There's a whole range of shades in between. And that's a result of the type of genes that are actually interacting here. So you don't have one particular, or rather 2 contrasts. You have a continuum of characteristics because of the many genes that control it. Depending where you are on the spectrum, if you have the dominant, you would be dark, and moving towards the recessive, you would be on the lighter color. And if you have an absent of some of the genes that are there, then you would be an albino, and that's how it goes. By the way, height is also controlled in the same way. I'm hoping that clears that up for you. I just want to complete this question. The question said we must also give the phenotypes of this. So the phenotype here would be 50% that are brown and 50% that are black. Gray, sorry, in this case. The other one was gray, not black. And that would be your complete answer. Notice, guys, that you're getting marks for saying P1 and fertilization. You get it, and F1. At the bottom here, you'll have F1 here. You get a mark for that. And meiosis and fertilization gives you another mark. I've put this here, and as we're going to be doing problems, we're going to use the same sample all the time, so that you get used to that format all the time. They say in humans, the gene for curly hair is dominant over that for straight hair. Use the letter H, capital H, to represent the gene for curly hair, and the letter small h, lowercase, to represent for straight hair. Okay? Explain why the statement below is true. A person with curly hair may be heterozygous or homozygous for this characteristic. Where does this come from, guys? It comes from the rule I told you just now. When the cat is away, the mice will play. So if curly hair is dominant, then curly hair can be either that homozygous, or it can be heterozygous. This is still curly hair. Although this parent or this individual has the gene for straight hair, the cat is around, the strong gene, the dominant one is there. And as long as the dominant gene is there, it masks or shadows the recessive characteristics. This is Mendel's law of dominance. And we also see in the previous slide, Mendel's law of segregation, that when meiosis takes place, the genes controlling a characteristic separate into different gametes. Now you must link this to your meiosis. This is what happens in meiosis. The homologous chromosomes separate, and the individual gametes have only half the chromosome number. That means they have half the genes. And this is what's happening here. And when fertilization takes place, then this sperm fertilizes that egg. The genes come together from the mother and the father. And that's why we join them up here, or we put them in a Punnett square. This is what you must have in your mind when you're answering this question. During sperm and egg production, they separate. During fertilization, they join. Not the same one, obviously, because it's not self-fertilization. We're talking of sexual reproduction where individuals are exchanging sperms, basically, and fertilization takes place. So let's go back to the next one we were dealing with. So that's why we can say that is the answer there. In humans, the gene, we said that already, show diagrammatically by a genetic cross how a man with curly hair who marries a woman with curly hair may have a child with straight hair. Oh, man is curly hair, woman is curly hair, the child is straight hair. Is there foul play here? No. It is possible because of the previous question. What did we say about curly-haired parents? They may be heterozygous. So that was a carry on for this one here. Now they're telling you that the child has straight hair. So in other words, one of the children here, when fertilization takes place, must be H, too small h. To have straight hair, this child must have that. They told you this, because straight hair is recessive, and it will only show itself in the homozygous condition. So that means, where did this child get the small h and the small h from? It gets one from each parent. So in other words, we go back to the genotypes of the parents. Each of these parents must have the small h. Each of the parents must have the small h, because they gave it to the child. Unless, of course, there's some foul play taking place and somebody is interfering somewhere else. We won't get into that. We are assuming that this is now a closed situation where there's no tampering involved here. You know what I mean by tampering, right? Okay, so both parents needed to have the small h. But the problem also says that the man is curly-haired and the woman is curly-haired. So therefore, to have curly hair, they had to have the dominant gene as well. So that already sorts our genotype for the parents. And they already told us the phenotype. They are curly. Both are curly. It's given to you in the question. Now, we've got to go to the meiosis takes place, and hence, we'll have H and another one H, small one now. And the same thing is going to happen on this side, and another one there. And I'm not going to cross this way. I'm going to go straight to the Punnett square. I prefer you use the Punnett square because the Punnett square is neater and leads to less confusion. So you take whatever is on the left, keep it on the left. Whatever is on the right, put it on top. By the way, you can switch these around, but you've got to keep the like with each other. I use this so that you don't get confused. Whatever is on the left, keep it on the left. Whatever is on the right, keep it on top. Because it's a multiplication situation. So whether you say h times h times h times h or 4 times 5, 5 times 4, doesn't make a difference. Okay? Now we cross. Let's see how easy it is in the square. H and H, two capital H's, so we write it down. That one and that one, you get this one gives you a capital H, that one gives you a small h. This one and that one gives you a small h and a big H, and two small h's. That's that possibility. That is why it is possible, there's a 25% chance every time fertilization takes place that these two curly-haired parents can have a straight-haired child. I'm hoping you're with me on that. Remember, it doesn't mean this will be the first child, that's the second, third, fourth. No. It means every time fertilization takes place, these are the possibilities. These are the probabilities. This is what genetics is all about. It's telling you about probabilities. And you're working it out using the Punnett square or the line diagram here. Phenotype, we given that already. We didn't ask for that. We just showed Y. They say here now, show the diagram below, which shows 3 generations of snapdragon plants, and answer the questions. Using the following symbols for the contrasting alleles. W, capital W, for white flowers, and R, capital R, for red flowers. Gary, there's a clue given in this question. The fact that they used a capital W and a capital R, 2 capital letters, tells you an important thing. It means that something here about dominance is not as we normally do it, because in a complete dominant situation, we'll have a capital letter and a small letter. They don't always have to give you this, though. They could have given you the results, and you had to work out backwards as well. So they give you the diagram. It's also a different diagram to show you almost a pedigree diagram of sorts. They're telling you red flowers is the white one there, white flowers is the blank in this case here, and those that are shaded are pink. Earlier we talked about codominance, where both are equally represented. Now we are talking of a situation where they're not equally represented. They had red, white, but from them we're getting pink, a new color. This, for a start, is incomplete dominance. So in incomplete dominance, the offspring that are produced will have a blend of the two, a new color, a new characteristic. Whereas in codominance, both genes are equally expressed. For example, black with white spots, or white with black spots. Or in the blood groups, if we take A and B and we put them together, we get blood group AB. A is dominant, B is also dominant. So they are co-dominant. That's what we are showing here. So first of all, we must understand that this is, in this case here, we're talking about incomplete dominance. State the kind of dominance shown here. Because of that pink, it becomes incomplete. Use the symbols R and W and write down the genotypes of each of the following snapdragon plants, A, B, and C. I'm going to go straight to the diagram. A was red. So red will be 2 capital R's. B was white. It'll be 2 capital W's. Please make sure that your capital W looks like a capital W. You don't make one look like that and one look like that. Already you're showing one is capital, one is small, and that's what they don't want to see. They want to see that they're equal, but capital letters, because it's incomplete dominance. And C, in this case here, will still be capital W, because it is not a blend of the two. It's the same as that one there. That one will be capital R, and this one becomes RW. Capital R, capital W. You've got to show the 2 capital letters. Why? Because you're showing that neither of the two is dominant to the other one. Important there. All right? We've done that one. It is possible to trace the inheritance of characteristics such as blood groups. Now we're looking at blood groups and genetic disorders over a number of generations. The pedigree diagram below shows the blood groups of individuals of a family. The blood groups are indicated inside the circle or the square. The blood groups of individuals W and X are not indicated. So they've given you some, and W and X they didn't give you. So they're telling you that this is blood group O here. That child from this relationship is blood group A, and another child is blood group B. Okay? They're also telling you here that this is another child from this blood group A is blood group O. Now, to understand this, guys, they want to know all the possible genotypes of individuals W. W is this one here. Now, what did we get from here? From these 2 parents, we got a B and we got an A. The one parent was O. And if you know your work, you'll know that blood group O is recessive. And we represent it with 2 small i's. Blood group A can either be I, capital A, capital I, capital A, homozygous, or capital IA with a small I. That means it is heterozygous. B, same as A, but with the B replacing the A's. O is that. And AB, IA, IB, codominance. They both equally dominant and they both showing up. Now, if this person here is blood group O, Now watch what I do, because this is a pedigree diagram and blood groups in one question, guys. So if that's blood group O, it's got to have two small i's, because when the cat is away, the mice will play, and O is recessive. So in other words, these children, 1 and 2, must have a small I. They must have a small I. They got from this mother. But this one is blood group B. How does it become B? It has to have a I with a capital B to become that. So where would it get it from? Can it get it from this one? No, this one doesn't have it. So it had to get it from this parent here. And this one is blood group A. How does it become blood group A? Because it can only be that way because of this one here. There, and that one there. And where would it get the A from? There you go. So the parent W had to be IA, IB. It can't be anything else. Because this child is A, that one is B, unless they were hanky-panky. We're saying that's not happening. And they want to know about X. Here's a child that's got blood group O. This one here was blood group A. So this could be IAI or IAIA. But we're saying it got this from the mother, so it cannot be that. So if this one got that one from there, it must get the other one from the other parent there. And this parent could have been either that, because we don't have any further evidence, or it could have been that, because from that I and that I you're getting blood group O there, or it could have even been IBI. So the person could have been BI, AI, or two small i's. I'm hoping you're with me, guys. I'm hoping you're with me. I'm hoping you're with me. Notice in a pedigree diagram, females are usually drawn in circles and males in squares. In a problem situation, they would shade those that are affected, and those that are unaffected are unshaded. Now we've given all the genotypes on the diagram there. With that, over to you, sir.
All right. Wow. Aslam, I can't even tell you how much I'm loving the action on the page. You guys keep on posting. Let me know what you guys are thinking. Nintendo, I would give you my t-shirt, but this is the only one that I have. But I'm going to make a plan to try and get you your own. But anyway, mindsetters, keep posting, keep chatting on the page, keep letting us know what you guys are thinking. If you're lost and if you need help, make sure you post. But we'll see you after this break. And welcome back, mindsetters. Hope you had a nice little break there, and you're back, you're ready, you're good to go. You've got your pens, pads out, and you're making notes, because I know a lot of you are writing exams really soon. So make sure you're paying attention. And on paying attention, just want to say, wow, guys, I just want to give you guys a thumbs up. Thank you so much for posting, making my job a lot easier. Yes. Before we continue, Aslam, I actually have a couple of questions here from the mindsetters. We have one here from Kashifa, who's a regular, who's been missing for like forever. But anyway, she's back on the page, that's all that matters. She wanted to find out, where does P1 end?
Okay, Kashifa, welcome back, Kashifa. Now you know that all your other insignificant papers are over, and the main one is on Friday. Life science. Remember, it's life science. Okay, you want to know about P1. Remember, P1, what does it tell you? It's actually telling you P1 is the first set of parents in a problem. So if they say a particular a brown dog mates with a white dog, and they have these offspring, and then these offspring mate again, and they have a second set of offspring. Then they will tell you, give the result till the F2 generation. So you start off with your P1, which will be your brown that we spoke about earlier. In this case here, these are your F1, your P1, your parents, the first set of parents. So it ends right there. That is your parent, and that's where it ends. It stays there. Now, if you follow through with this problem, and if they told you now, if this one and a homozygous for curly hair mate, what will be the chances of them getting X, Y, or Z? Then these two now will become your P2. And your result will be your F2. And so it goes. Generally, for our life science, we only go up to F2. You can go further, obviously, but in our studies for our exams, it generally goes to F2. So P1 is just there. That's where it ends. That's your parents, your first parents, and it ends there. Your F1 will be here at the bottom, because this is your result. Next question.
All right, then we had another one that came in from Nhlakanipo. I hope I said that right. Nhlakanipo. He wanted to find out, hey Ty, can you please ask Aslam if we are always going to be informed whether the disorder we are going to be dealing with in the examination question, or is it dominant or recessive before we cross, or should we know them by heart?
Firstly, you need to know the ones that we have dealt with, whether they're dominant or recessive, you need to know that. But generally in a question, either they would tell you that one is dominant or the one is recessive, or if you pay careful attention, if you look at the offspring, that should tell you whether that disorder is dominant or recessive as well. So you need to read the question. Obviously, if we give you a disorder that you haven't done, it's not prescribed in Circular E16 of 2010, then obviously in that one we would have to tell you that the one is dominant or the one is recessive, or it must show in the question. But in terms of albinism, in terms of color blindness, in terms of hemophilia, you are expected to know that.
Okay, then we had another one that came in from Sifiso. He wanted to find out, is there a way of knowing from the statement when the F1 generation is going to result in incomplete dominance?
Okay, so in that question, let's just go back to that one there. That one there. Oh, I've gone past it already. Sorry, Sfiso. So after this one. Yeah, here we go. You got to look at the offspring to know whether it's incomplete or complete or co-dominant. Obviously, if in the bottom, your F1 generation, you see that the one is showing more than the others, then obviously you're talking about complete dominance. If they say, for example, we mated black rabbits with white rabbits and all the offspring were black, that tells you it's complete dominance. That means one was strong and one was weak. If they tell you we mated black rabbits and white rabbits and we got black rabbits with white spots, that tells you that this is co-dominant because they are both equally represented. And in the same example, if they said you had black rabbits and white rabbits and your offspring were all gray, that's incomplete dominance.
All right.
So that's where you got to look for that. Are those our questions?
That was all the questions so far, right?
Okay, keep them coming, guys. I'm glad you're asking that. Remember, the questions that have been asked here, many of you are thinking of the same questions, and these guys are helping you out by posting those questions. So we answer it, we clear everybody's minds of any confusion that they may have. Right, so let's move on. We're still on the blood groups. They say a woman has a child who is blood group B. Her child is blood group B. There are two men who are claiming paternity. Ooh, dangerous, dangerous. Two men are claiming paternity. What does that mean in our language? Two men are saying that they're the father of this child from this one woman. Dangerous. Okay, Tebogo, one of the is blood group O, and Imran is blood group AB. The woman's blood group is A. Okay, so let's look at what we have here. The child is blood group B. Let me do this at the bottom, it'll be easier to see. The child is blood group B. Tebogo, T I'm going to write there, is blood group O. Imran is blood group AB, Imran, I for Imran, AB. And the woman, we're not saying her name because there's two people involved and we don't want to shame and blame her here on TV. So her blood group is A. All right? Now, we've done something very important earlier. If a person is B, he or she can either be IB, IB, or IBI. That means heterozygous or homozygous. O can only be homozygous because it's a recessive. AB, because they are co-dominant, it will be IA, IB. And blood group A can either be IA, IA, or IAI. There we have it, IAI. Now the child was blood group B, okay? The woman was blood group B, A, all right? The fact that this child is blood group B already tells you that this woman can't be IAIA. Because if she was IAIA, the child had to have one A segment in his blood. So he would have had to be IA or AB, in other words. So this is what the woman actually is in this example, firstly. That's our first thing that we have to look at. Then we go further, we look at these two gentlemen. Teboho is blood group O. The woman didn't have one spot of B in her blood. This child's got B. Could Teboho have given this B? No, he can't because he doesn't have it. He has only these two small eyes, which is blood group O. So he's out. Even without even looking at the problem, he's out already. So it can only be Imran. I could be funny and say, look at the race of the child, because if you live in South Africa and you're stereotyping everybody, you say Imran, Indian. Teboho, black. What is the child? Colored. Black. You could have made a decision on that, but that's not how we do things, guys. Get out of the stereotype, because Imran can be anybody, any color. Teboho can be any color as well. We're living in a multiracial, multicultural society, so let's get rid of those stereotypes. But we base our stuff on scientific explanations. And here's the scientific explanation, and we could prove that there. But remember, in a blood group type, in this situation here, all we prove in a situation like this, that Imran could be the father. I could be blood group B. Tai could be blood group B. So it could be anyone outside of these two that could have also been the father. So all we are saying is that from these two, Teboho definitely is not the father. We can do that. What's good about this, we can prove when you're not the father. Well, sometimes it may not be good if you want to be the father, okay? So that's another story, okay? So that's where we're looking at there. Who can claim paternity of the child, Teboho or Imran? And we have shown there that Imran rather, the right way to pronounce it, Imran. My accounting teachers used to say Himran. That's why I'm saying that now, just because I'm thinking fondly, by the way, of her in the show here. So we have to show all the working. I have shown you in previous slides how to work out, so I'm not going to go through the working out. In this particular diagram, already just by looking at this, can you see how important this homework is? When you're answering a question, how important is it to do this homework? Because this homework guides what you're going to go forward. And then you're going to just put this one there, Imran, and you're going to put that to show that when this gamete fuses with that gamete, that is how we got this child possibility, the possibility of that child being blood group B. So you're going to show it in a diagrammatic form, P1, phenotype, genotype, meiosis, gametes, fertilization, F1. You're going to show all those steps, because even if your answer is wrong, you're going to get at least 2 marks for saying P1 and F1. For saying meiosis and fertilization. There's 2 marks already out of 6. That's 1/3, 33%. You already passed this problem. So keep that in mind when you're doing that. Okay, so we have covered that. We're not going to do the problem. Now they want to know if a person's blood group is B positive. What does the positive mean? What does that positive sign mean? It means that this person is positive for what we call the rhesus factor. Why is it called a rhesus factor? Because it was found first in monkeys called the rhesus monkeys. This factor, if you have it, you are positive. If you don't have it, you are negative. That is why our blood groups are not just A, B, O, and AB. It's A positive, A negative, B positive, B negative, and so on. So if you have it, you're positive. If you don't have it, you're negative. There are complications in several pregnancies over several times if the father is positive and the mother is negative. You can read a little bit about that. I do not want to go into detail of that right now. We are more interested in how we determine blood groups and how you'll notice the way we set out a problem for blood groups, the way we set a problem for codominance, for incomplete dominance, for any disease is the same. It doesn't change. The set out is still the same. The format is the same. So keep that in mind. Now they're saying a hemophiliac man is married to a non-hemophiliac woman. They have 3 children, 2 sons who are non-hemophilic and a daughter who is hemophiliac. Okay? 2 sons, non-hemophilic, and a daughter who is hemophiliac. The mother is expecting another boy. What are the chances that this 3rd son will be hemophiliac? Explain your answer using a genetic diagram to illustrate your answer. Now, guys, this is important because this one here, although the format is the same, the the way that we use, the letters that we use are slightly different. Because hemophilia is a sex-linked disease. You have to know this because you have to learn hemophilia. It's a sex-linked disease. That means, what does this mean? It's a disease that is carried on the sex chromosomes. In other words, it's carried on the X chromosome because the Y chromosome is too small to carry any disorders. The X chromosome is larger of the two. And remember, females are XX and males are XY. To be a male, the male has to have the Y chromosome. And Ty, if you look at that board, the male can only contribute to the maleness of his children. So if you get married, Ty, and if you're getting only daughters, it's your fault. Okay? So getting
Fair
another
enough.
wife is not going to solve the problem. Okay? Because your Y chromosome is determining whether that child is a male or a female. It means your Y chromosomes are on sperms that are swimming slightly slower. Okay? So you need to have some Red Bull. Okay, right. So this one here, now they want to know, the mother is expecting another boy. First of all, the fact, guys, that they had a daughter who is hemophiliac. Hemophiliac, by the way, hemophilia is recessive disorder. It's a recessive disorder. And we use either XH or X small h in this answer. The fact that a daughter is hemophiliac, that means she had to have two small x, X small h, recessive character, homozygous. So if she had that, that means both the parents had to have the small h. And they're saying a hemophiliac man, okay, that's easy, and non-hemophiliac woman. She's not hemophiliac, but the daughter has both, so she had to have it. She's not hemophiliac, but let's explain why. First of all, the father, XHY. Because there's nothing on the Y, there's no other one on the Y. The fact that he has only one already makes him hemophiliac because he does not have another gene to counteract this one on the Y. The mother, they're saying she's not hemophiliac, but her daughter is hemophiliac. That means she had to have one small one. So in other words, guys, what it means is, sorry, I'm writing this in the wrong place there. That should be at the bottom here. I'm just putting arrows there to put that there. That's the genotype, not the phenotype. The phenotype, we'll just write it on the top. Phenotype, father, hemophiliac. Mother, normal. Because if we look at this woman, we won't be able to say from looking at her outside whether she's hemophiliac or a carrier. A carrier you can't see because they're not bleeding. They have the gene, but you can't see it. So they, in our eyes, they are normal. So she's normal, and this is what she is, but she's a carrier. You cannot write under phenotype, and we had a big debate in a memo discussion last year about this, and we all understood why. Because when we look at this individual, we cannot say she's a carrier. It doesn't make her look any different from anybody else. It's only in the genotype that she's a carrier. Phenotype, she's still normal. Right? So you talk of a hemophiliac or normal. You don't talk of a carrier under phenotype. So that's our genotype of the parents. Meiosis takes place, we separate them. Look, everything else is exactly the same, guys. There's the XH and there's the Y. Here's the X capital H and here's the X small h. And I'm not using the lines as I told you earlier. I'm using the Punnett square, so it makes it easier and faster as well for us here. Whatever is on the left, keep it on the left. X, H, Y. Whatever is on the right, put it on top. X, H, X small h. Now everything else is the same. We only put in, the only difficulty here is to put that X and Y. Let me make this X nice because otherwise it looks like a Y. Remember, females have two X. X, H, X small h, X big H, X small h, X small h, Y, X big H, Y, X small h. They want to know how come they got, what is the chance of this third son being a hemophiliac? How many children here? 4. Out of these 4, what is the chance of this son being hemophiliac? There are 2 sons here, 1, 2. If you take all the children, then it's a 25% chance. But because we're talking about sons, we can't take the daughters in this whole equation. Out of 2 sons, 1 can be hemophiliac, 1 is normal. So therefore, 1 out of 2, half, or 50%. Or we can even say 1 is to 1. Because for every 1 hemophiliac, there's a chance of 1 being a non-hemophiliac. I'm hoping you understand that. And when we're writing the phenotype, if we had to do this whole problem, we must write here, for example, what's this? This is a girl, right? So girl, female, F, I'm writing female, normal. Although she's got the small h, but she's got the big H as well, so she's normal. The other female is hemophiliac. The others are males. Male, normal, 1, 1, 1, and 1 male hemophiliac. This is important to get this. This is only 1 mark to write all of this, but you have to say female, normal, female hemophiliac, male, normal, male hemophiliac. Otherwise, you do not get that mark, because the phenotype is either they're males or females. Good. This question, guys, I hope you realize that we have dealt with problems with blood groups, genetic disorders, sex-linked, because the ordinary disorders are just like a normal genetic cross. The only difference with the hemophilia is that you have to use X and Y. This question here, very easy question, says study the diagram below and answer the questions that follow. This deals with karyotype. They've given you all the chromosome map here, and they're telling you karyotype of a person. There's all 23 sets of chromosomes here. Is this karyotype of a male or female? If you remember our earlier shows, we said that always go to the last pair, pair number 23. In this case, by the way, these are not X's. These are chromosomes. Understand that. These are chromosomes. So you can't say she got XX. Although that is true, but that's not what this stands for. Why do we say that she has XX? Because these 2 chromosomes are the same size. If this was XY, then the chromosomes would have been something like that and like that. They both still X because they're not X's. They are chromosomes, but one is bigger, one is smaller. Or if they just did this and that. Okay? I'm hoping you understand that. So in this diagram, you need to know whether it's a male or a female. That's the first thing. Where do you look? Here. Then you want to know whether this person has Down syndrome or not. You look here. Chromosome number 21. If there are 2, each one, you see there's only 2, 2, 2, 2. This one has 3. It has an extra chromosome on chromosome number 21. That means this person has Down's syndrome. That is how we use this diagram. Those are the important things about the karyotype or karyogram, to see this, and also for genetic counseling to be able to tell the people what we expect from what's good the offspring and what to deal with that as well. Right? How many chromosomes does this person have? 47 chromosomes. Can you see that? Good, because everyone, 23 times 2 is 46, plus the 1 gives you 47. Guys, that's all we have time for today. Please work hard and go smash that paper on Friday.
All right, on that note, wow, there's so much information crammed in. I'm sorry if I didn't get a chance to get to all your questions, but guys, doesn't mean that you stop posting. Make sure that you keep talking to me, keep posting on the page, keep talking to each other as mindsetters. Mindsetters, if you've seen those questions and you think you can help, give it a shot, give it a whirl. You don't know, you could actually help out someone for this Friday exam, which they could pass because of you helping them out. But this is where I, as Ty, sign off and say thank you and see you next time. Cheers.